Hello, I'm Tim Sandle, and I'd like to welcome you to the latest 5 minutes video. And this is the seventh video in the series so far. And like every other video, these videos are dealing with one topic, making no more than 3 points, and ideally running for no more than 5 minutes. So today's subject is the humble agar plate, or more specifically, the settle plate. I'm sure you're very familiar with the design of these devices. Now, the settle plate is an item of microbiological culture media. Now, in order to grow and reproduce microorganisms, the microorganisms require certain biochemical factors, nutrients and sources of energy, and they also need certain biophysical factors, which is the ambient conditions, the pH, the right atmosphere and the right temperature to grow. Now, the first point I'd like to make is just about culture media in general. So in the natural environment, microorganisms have adapted over millions of years to grow and thrive in habitats that are suitable to their needs. And to replicate these conditions in the laboratory, then we need to develop specialised culture media, which can be a broth or like an agar plate that contain the necessary nutrients to promote microbial growth. Now, most of you will be too young to remember a television botanist called David Bellamy. He used to become very excited about the constituents of plants. And I try and hold back, but just to run through that, there's a whole range of nutrient and physical requirements for microbial growth. And these include water, a source of energy, sources of carbon like glucose or nitrogen from amino acids, sulfur, phosphorus, minerals, metals, buffer salts, vitamins, and a whole array of growth factors. And in this plate contains all of those things I've listed. And the main source of these growth factors comes from tryptophan, and tryptophan is derived from cows. And this link to animal byproducts means that it's the way that most commercial culture media companies arose was using the side products of the food industry, specifically making meat products, as a way of utilising the leftover substances. And making media is a little bit like cookery. You need to throw in the right ingredients in order to get the right balance, in order to grow the most common microorganisms. And the ones that we're most interested in are the ones that come from the human body or from the outside environment. And the crucial element to this is the solid medium. And this is the case of the agar. The agar provides the solid base. And agar is derived from seaweed. And for these types of agar plates, then it's a red seaweed that's used to create the jelly-like medium and gives it the beige appearance. And agar's been around since the late 19th century, formed from the laboratory of a German bacteriologist called Robert Koch, and he was a pioneer in research into tuberculosis. All right, that's very interesting. But what does it mean for what you're doing in the filling area? Well, the second point is about the plate, what the plate does, and why being positioned in a certain area is so important. So a settle plate at grade A is designed to inform us about contamination that might fall into an open vial. So what we recover on a plate might indicate what may have gone into a vial. And this is either through being transported in the airstream or through gravitational settling. And we try and avoid the airstream thing through the unidirectional airflow design, where we're trying to blow away from the vials. The gravitational settling is often more of a factor that could arise during an intervention. And when you're doing an intervention and you get close to another object, like conveyor belt or vial or the plate itself, the closer two surfaces become to each other, the slower air becomes, and the greater the chance is of things depositing or settling, hence the name saddle plate, from the air onto the surface of the plate, and turbulence can also exacerbate that. My third and last point is about labelling. So it's important to label plates as they go in, and to do this as close to the time of the activity as possible, because once plates are removed from their packets, then you do have risks of lids becoming loose or plates becoming contaminated. It's also really important to label plates with the correct information as well, because the location matters. Some locations present a bigger risk than others, and also we never want to get grade A and grade B confused, because if we get those confused, then we can get the wrong contamination and go down the completely wrong way. It's also very important to avoid touching the plate and making sure that the lid is secure. And then for transport, it's important that plates go into plate racks, or they're secured by 2 sets of rubber bands to avoid contamination occurring, and particularly with the lid becoming loose. Now, as you can imagine, there's loads I can keep on saying about plates, but I'll save that for another time in a future video in the series. So this has been about saddle plates. So goodbye for now. Just a reminder, I'm Tim Sandle, BPL site microbiologist, available to answer questions at any time, so please email me. And have a good shift and keep on staying safe. Till next time, goodbye.